Okay, so we looked at the six different types of membrane proteins. So your responsibility is to be able to name all of those and to be able to explain how they function. And then the application part of that, right, that's really just memorization, right? And now we're going to work a little bit to apply that information. And the application comes in looking at specific diseases, and then based on an explanation of the diseases, I want you to go back and figure out what type of membrane protein is defective in a particular disorder. So the first one is Duchenne's muscular dystrophy. So what I want you to do is pause here and then read through this and then find the information that tells you what type of the 6 membrane proteins is affected in Duchenne's muscular dystrophy. So pause, and then I'll come back and kind of give you a hint if you need it, and then I'll tell you the answer. So hopefully you were able to read through that and find that, or just in case you didn't, so I'll highlight the section that will give you the information. And highlighting kind of covers over it, so I'm sorry. But I will read that to you. But it is in this section where, so the dystrophin protein has a major structural role in muscle because it links the internal cytoskeleton to the extracellular matrix. That is where your answer is. And then all of the other stuff is just more information about the particular disorder. So based on that highlight there, now can you tell me what type of membrane protein is affected? So I'll give you a pause to think about it, and maybe if you need to, go back and see which one of those links is important in linking the internal cytoskeleton to the extracellular matrix. So hopefully what you were able to determine is that dystrophin is an attachment protein. And when you don't have the proper function of the dystrophin, if you don't have that protein, then you don't have proper cell structure. And now in this one, dystrophin is specifically related to muscle. And so the muscles are not able to function appropriately because they don't have the specific dystrophin protein, and they will not function properly. So this is where I start to introduce you to how a protein mutation or malfunction is going to lead to particular disorders, because the end project for the semester is to pick a disease that you're interested in, and then I will share with you a primary research article that relates that disease to a specific protein mutation. And this is your baby intro into that. We will start to define the groups for those coming up in, I don't think this next class, but one of the next ones coming up. And then your job as a group is to pick a disease that is interesting to you, and then I will help you find a primary research article that relates it to a specific mutation or malfunction of a protein. So these are just examples of possible ones that you could choose. So that is Duchenne's muscular dystrophy. The next one is cystic fibrosis. And so for this one, again, read through and then see which one of the 6 membrane protein types is defective in cystic fibrosis. So pause, read through, and take your best guess. And this one, hopefully it was a little bit easier. You didn't have to read too far. You can see that this right here, so it's a transporter class ion channel that transports chloride and thiocyanate ions across epithelial cell membranes. So if you didn't get it already, read that particular part of the sentence without all of the other confusing stuff, and then come back and look at your membrane proteins and see if you can pick which one is defective. So if you got from that, when it says transport here, that the cystic fibrosis transmembrane conductance regulator is a transport protein. It is specific to chloride and some other anions, but so negative things are going to move across the membrane. It does take some energy, but instead of moving the chloride against the concentration gradient, it's using energy to move more chloride out that's moving along the concentration gradient. So it's a little bit confusing, and it's just one of those specific transporters that is using energy to move things faster along the concentration gradient instead of using energy to move it against the concentration gradient. Not super important here. Just remember that it's a transport protein and allows chloride to move out of the cell. So it does the way it functions gets a little bit complicated. So I'm going to diagram it out as best I can here. And I think it's best to just using this diagram instead of having me draw it. Generally seems to be the case. But what I want you to focus on for this is that, so these are the epithelial linings of the lungs, and then out, so this is inside of this cell, this is inside of this cell, but each cell is going to have all of these things. But the way it is going to happen is that with the CFTR, the cystic fibrosis transmembrane regulator, right, so this one right here, it is pumping that chloride out. And when the chloride pumps out, it actually keeps the sodium out here. And so that means that water is going to stay in that extracellular space. So chloride coming out is attracting, chloride coming out is keeping the sodium in here as well. And if you have concentrations of sodium and chloride more higher out here, then that it's not salt form, but those ions that form the salt, right? Those are going to promote water moving out of the cell and keeping it in the extracellular space. And then the idea is that if there is mucus that builds up, the water thins the mucus and allows it to be coughed out. However, in individuals with cystic fibrosis, in some forms of cystic fibrosis, this transmembrane receptor that normally pumps the chloride out is not functional. And so now the chloride doesn't move across the membrane, and then that means that the sodium's not attracted to stay out. So you can see that there is more sodium coming back in. So that means there's less water staying in that extracellular space. So less water is moving out, being attracted by the chloride, and then more sodium's going in, and so more water is going back in. And so there is less water in the extracellular space, and so that keeps the mucus thick, right? If you've ever been sick and you cough up something really gross, that's in your lungs. Yuck. And that stays in your lungs if the water is not in that extracellular space to help clear it out. And so for cystic fibrosis, what normally happens is that you have this mucus that doesn't get cleared out, and now you have bacterial infections that are more likely to happen within the lungs. So in general, the CFTR membrane protein, which is a transport protein, normally just all it's doing is pumping chloride ions across the membrane. That is it. But if it doesn't work, that simple function can cause a really severe side effect. The individuals with cystic fibrosis, the prognosis is way better than it was before, but there are still complications that can happen and infections that can occur that will kill somebody. But now they do have medications that actually increase the function of this receptor. It used to be that all you could do was just pound on the back of somebody to cause that mechanical disruption of the mucus. But now they have like jackets that they can wear that kind of compress then there now is more medication that can combat it as well. So all it is that chloride ion not moving across, and then the water is not staying in the extracellular space to clear out the mucus. And then our last membrane protein is a growth hormone deficiency. So some of them are dwarfism, and then generally just short stature because of a defect in a specific membrane protein. So read through this, see if you can find the part in there that tells you which type of membrane protein is altered. So pause and give it a try. So hopefully you're able to read through and then see that the growth hormone releasing hormone receptor is a G protein coupled receptor that binds the growth hormone releasing hormone, and then as a result of that, the growth hormone gets released. But in the growth hormone deficiency, there is a defect in this growth hormone releasing hormone receptor. So I know it says it there, but go ahead and pause and see if you need to. Go ahead and see which one of those types of membrane proteins is defective. And just like it says here, that growth hormone releasing hormone receptor is a G protein coupled receptor. So again, I'll go through and give you the specifics of it and do my best to explain it. I love when I find a diagram and I don't have to draw things. So in this one, the normal signaling is you have this growth hormone releasing hormone, right? It binds to this growth hormone releasing hormone receptor, which is a G protein coupled receptor, which can activate this G protein that happens to be termed the GS alpha protein. That activates adenylyl cyclase, which we actually saw before, which is the enzyme that converts the ATP to cyclic AMP. And then that cyclic AMP is able to lead to the release of growth hormone. And then that growth hormone release leads to cell growth. But in individuals with growth hormone deficiency, right, this is defective. So even if that growth hormone releasing hormone is present, it is not going to activate the receptor, which is the G protein coupled receptor. So this isn't going to happen. And then this enzyme is not going to be activated. So that cyclic AMP can't be activated to open the calcium channels to cause the growth hormone release. So as a result, there's going to be less cell growth. And specifically, it generally, the biggest effect is in bone growth, because that is what allows, really bone growth is the biggest part of stature. And so all of this is just because there is a mutation in the one particular G protein coupled receptor. So again, tying a mutation of a protein, that protein does not have the proper shape or does not have the proper function, or in some cases just not even there, then you don't get the cellular response that it is supposed to do, and then you don't get the target or the change in the cell function that you are normally supposed to get. So what I want you to start thinking about is, like, what type of generally genetic disorder are you interested in? And then I will have a whole list of them that you and your groups can choose from coming up. But just want to highlight that the function of every single protein in your body is important to happen correctly, or that is when diseases and disorders develop. Most of it stems from the dysfunctional protein structure and protein function. And also, there are kind of bigger picture things, what we call epigenetic changes, where you have the regular DNA sequence, but then there things have come on, like methylation of the DNA has happened, so it changes the expression of those proteins as well. So they're not only just at the basic DNA sequence level, but at the altered ability of that DNA to be expressed and to get that protein expression. It's kind of amazing. And there are many things that come into play to affect that. So stress, environment can all lead to epigenetic changes. And some of those epigenetic changes can be transgenerational. So the environment that your grandparents grew up in can have affected how your proteins are being expressed today. It's amazing. Yay, biology.